Imagine your DNA as a massive library, with each book representing a gene. These books contain information that makes you who you are. However, not every book is available to read. Some of them are open, ready for anybody to read, while others are closed, keeping their information hidden. Epigenetics is the study of these differences, and controls whether a book in your DNA library is opened and read or kept closed. Take two identical twins for example. They both have the exact same DNA. However, the kind of life that they live can change their traits. One of them might exercise more often, while the other might have a healthier diet. This could lead to them developing different traits or even different genetic diseases. If they have the exact same DNA, how can this happen? This is where epigenetics comes into play. Epigenetics is able to turn on and off genes through the use of epigenetic marks. One example of these marks is something called methyl groups. In a process called DNA methylation, methyl groups can attach to certain parts of DNA, which prevents gene expression from occurring. Epigenetic marks can also be attached to histones in a process called histone modification. Histones are the proteins that DNA is wrapped around in order for it to fit inside of a cell. Epigenetic marks can control how tightly DNA is wrapped around these histones, making it easier or harder for gene expression to occur. Epigenetics is an extremely important field of research, as having a better understanding of it can help us find new ways to combat diseases. For example, oftentimes cancer cells' epigenetic marks aren't placed properly, with some genes that are supposed to be turned on turned off, and vice versa. In a new field called epigenetic therapy, scientists are trying to find ways to combat cancer and other diseases by restoring the correct patterns of epigenetic marks. By understanding epigenetics, we can become librarians of our own DNA, opening the books that we want and closing the ones that cause harm.